Good morning. Can everyone hear me? I'm going to stand because I'm a restless creature. But Your Majesty, Ms. Queen Sylvia, very nice having you here. Thanks very much for having me in the Vatican again. It's an extraordinary honor to be here and an opportunity to discuss the concept of addiction. As I was listening yesterday and also in our talks at lunch and dinner, it became evident that there's an enormous amount of interest to try to understand the changes in policies that are happening around marijuana. So at the last minute, I changed my presentation to be able to accommodate for this interest. So what I'm going to be speaking today is what is it the neuroscience on what we know about the effects of marijuana in the human brain. How do I change the next slide? Okay, that makes a difference. We now, in the election, there was not much attention paid to the fact that now we have 4 new states that have approved the recreational legal marijuana. So we've gone from 4 states to 8 states plus Washington, D.C., where the recreational use of marijuana is accepted. And then in darker green, you see all of the states that have approved medical marijuana. So as of now, we have more than 28 states plus DC where marijuana is legal, either for recreation or medical purposes. That's more than half of the United States. Now, what are the consequences? And I think that we need to look at it in a very critical way to try to understand and do interventions that can minimize the likelihood of adverse effects. You see there the scores of regular use of marijuana, and I chose this metric because to me, the regular use of marijuana, which is the use of marijuana on a daily basis or close to daily basis, is the one that's most likely associated with adverse effects. One of the things that I do want to commend, because it's very relevant for us, is that among teenagers that you see that in sort of grayish symbols, we have not seen an increase in the regular use of marijuana, which has remained elevated if you think about it in overall context. Among 12th graders, 6% of them use marijuana regularly. But I do want you to pay attention to the graph in dark green, which is 18 to 25, where we see the highest rates of use of marijuana. And there we have, think about it, 9% of our 18 to 25-year-old smoking marijuana regularly. That's close to 1 in 10. And that is a stage in our lives when we're going in the transition from going to school to becoming a productive member of the society. And it's at that stage of great challenges that we're seeing the greatest consumption of marijuana. And I will come to that because the question that follows is what could be potential negative consequences of regular use of marijuana in an adult brain. The other very important fact that we need to consider as we are addressing the changes in policy is that the marijuana that is available for consumption is very different from the marijuana that existed in the past that accounted for, that was also the one that was used to make most of the scientific studies that had enabled us to guide policy. In particular, I want to concentrate on the content of THC in red there, because this is the active ingredient of marijuana. It is this chemical component, that THC, that people seek in order to feel high, and it is the component that is associated with the addictive properties of marijuana. On the other hand, I want to commend the other one in blue, CBD or cannabidiol. This is the active ingredient that is most frequently responsible for the so-called medical properties of marijuana. For example, we know that cannabidiol is the chemical that's necessary for the therapeutic effects that are being shown for cannabinoids as it relates to epilepsy. And you can clearly see that the content of 9-THC that's responsible for the addictiveness and the psychoactive effects, that is the effects that affect the brain, are going dramatically up from 4% to 12%, and in many instances the content can go up to 25, 30%. And cannabidiol, which is the one with therapeutic properties, most of the therapeutic properties is going down. So this actually could account for the fact that we are seeing a significant increase throughout the United States of the number of medical emergencies associated with the use of marijuana, as well we have seen significant increases in people seeking treatment for marijuana addiction. Why do people take marijuana? Why do they take 9-THC? 9-THC is a cannabinoid, and we produce our own endogenous cannabinoids. Cannabinoids are the most widely investigated, which we call endogenous cannabinoids, anandamide and 2-AG. And these are chemical substances that are widely distributed in our brain and in our body to regulate chemical signaling in the brain. And I would venture to say that endogenous cannabinoids basically regulate almost every process in the brain in ways that are very different from the classical neurotransmitters. What do I mean by that? Normally, a neurotransmitter is a substance that is released from one cell to the other to signal back and therefore transmit that information. Endogenous cannabinoids are different because when a system is signaled, when there's excess signaling in the system, an enzyme produces the endogenous cannabinoids in situ, which then is liberated and acts backward to regulate this process. And that's exactly, and it does it by binding to receptors, cannabinoid 1 receptors and cannabinoid 2 receptors. And this is exactly the system that marijuana messes up with by directly interacting with these receptors. This is just to show the wide distribution of marijuana, of the cannabinoid 1 receptor, which is the one that's affected by THC. You see it in the brain, very high concentrations, but you also see it throughout the body. The other receptor, cannabinoid 2 receptor, very low concentrations in the brain, but extremely high concentrations throughout the body, particularly in cells that are involved in immunological responses. And this is the reason why there is interest for the potential of cannabinoids as therapeutic agents. That does not mean that marijuana itself is a medicine, but that active ingredients that are in marijuana can actually be tailored with chemically to produce active medications that could be beneficial in a wide variety of disorders. That would be like what we've done with many other chemical compounds that we took from our body and then improved them to maximize therapeutic and minimize side effects. This illustrates actually just what I was saying, that cannabinoid receptors actually are involved with the regulation of almost every function in the brain. And they are widely distributed, for example, in the reward areas in purple, and that's why they are addictive, but that's also why they make us feel good. Our own endogenous cannabinoids make us feel good. They regulate memory, consolidation, and formation, and that's why when people are intoxicated with marijuana, one of the processes that is disrupted is that of learning and memory, which can be very detrimental for teenagers or young people trying to go to school while at the same time being intoxicated with marijuana. In the cerebellum, where you see it in red, where it says coordination, that is an area also loaded with cannabinoid receptors that could explain to us why there is a significant increase and risk for car accidents when people smoke and drive at the same time. But almost, as I say, and I reiterate, almost every single process of the brain, every function is modulated by cannabinoids in the brain. And therefore, that's why we need to understand this as people become intoxicated and marijuana becomes more prevalent. Among the processes that are involved where cannabinoids get engaged with is the process of brain development. The brain is an extraordinarily complex organ that is formed, actually, if you were to ask me what is a simplified version, the most complex network that we can ever conceive of. The brains are cells interacting with one another in a multiplicity of networks that change dynamically as a function of the need and the process. And marijuana, evidently, by acting on these cannabinoid receptors that regulate that orchestration, is likely to have particularly a profound effect in the developing brain. Now, why is this of concern? This is of concern because among the medical properties of marijuana that are being advertised is that as an anti-nausea medication. And if you go into the web, and as you know, pregnancy, particularly during the first 3 months, is associated with nausea. And indeed, in the web, you can see advertisement of marijuana as a treatment for all of the ailments that may happen during pregnancy. We already know from studies that have been done in the independent investigators that women that smoke marijuana during pregnancy are much more likely to actually have infants of decreased birth weight, and that those infants are much more likely to require a neonatal intensive care unit. How important and how prevalent is the use of marijuana among pregnant women in the United States? We recently did an analysis that took information from 2002 to 2015 to try to actually get a prevalence estimate. And what we found is that while overall the prevalence rates of smoking in women that are pregnant is approximately 3% of the whole population of pregnant women in the United States, what is very notable is that among women that are on the first trimester, up to 6% of them have smoked marijuana. Now, why is this important? It is important because this is the first trimester, is the one where the fetal brain is the most susceptible to the adverse effects of drugs. And if you think about 6%, that's actually a very high number. What do we know in terms of how bad is marijuana for the fetal brain? We know that alcohol is very, very teratogenic, and it produces marked disruption in the brain of fetuses, of babies born out of mothers that drink. But we didn't know that about alcohol until 50 years ago. We have not been paying much attention from the epidemiological perspective about what are the consequences of smoking marijuana to the fetus once it's born. We know, and you see these images in colors, that actually show in the concentration of cannabinoid CB1 receptors. Very early on fetal development, they emerge. And as I say, their role is multiple, including forming connections between neurons, one neuron with the other, but also allowing for the neurons to migrate to the cortical areas. And those in animal models, we can see that exposure to 9-THC actually disrupts the number of neurons that end up in the cortex. Also, studies are starting to emerge in animal models that show, like the one in pink, you can see, and unfortunately I cannot point up in both of them, but you see the eyes of a normal animal, newborn animal, and this is an animal that was given a cannabinoid similar to marijuana, and you can clearly see the complete disruption on the facial structure of these animals. You see the eyes completely transformed. These are three different animals. The higher the dose, the worse the facial deformation of these animals, indicating that disruption of interactions of agonists like marijuana with the cannabinoid 1 receptors may have teratogenic effects. And this is something that as an institute, we're actually trying to address whether it is happening in humans or not. And as I say, I reiterate, there has not been sufficient attention paid to determine if this is the case in the humans or not. We've spoken a lot about the potential negative effects of the use of marijuana in the adolescent brain, because as we transition from childhood into adulthood, the brain actually is undergoing significant changes. And it's making changes that will allow to optimally perform in an environment that actually has many complex challenges. So that's why the brain is not fully formed until we are adult, because we do not know the environment that we're going to be actually exposed to. And during that developmental stage, the brain actually is like a sculpturing of your brain that is influenced by your environment. And drugs directly influence it. And in particular, cannabinoids, because they are involved itself with the architecture of the brain, are liable to produce significant changes during this period of time. We don't need sophisticated brain imaging technologies to realize that the use of marijuana does not seem to be associated in adolescence with good outcomes. In fact, independent studies have consistently shown us that kids that smoke marijuana during adolescence are much more likely to drop out of school, number one, but also to be unable to complete, if they keep on in school, to have good grades. The more frequent the use of marijuana, the worse the outcomes. The other thing that we've learned through all of these studies is that kids that smoke marijuana during adolescence are at much greater risk, number one, of becoming addicted to marijuana, but number two, of becoming addicted to other drugs independent of marijuana, which had led to the concept of marijuana acting as a gateway drug, a drug that if you get exposed as an adolescent will make you vulnerable to the addictive effects of other drugs of abuse. In addition to this, there has been some data, which is actually something that we cannot ignore, we need to look at it more carefully, that indicates that the consumption of marijuana during adolescence is associated with a higher risk for suicide attempts that cannot be understood in the context of an increase of risk for depression, because there's not such an increased risk of depression. But that may, in turn, identify the higher risk of impulsive behaviors associated with the consumption of marijuana that then may be driving this suicidal behavior associated with a frequent use of marijuana. What do we know about that important concept of marijuana as a gateway drug? It is, in humans, it's difficult to say, this person took marijuana and that's why they became addicted to cocaine or alcohol, because there may be the vulnerability that makes you vulnerable just in general to take drugs, and marijuana is the drug that's most easily accessible. But in animal models where we can actually manipulate that, studies have also more or less consistently shown that if you expose animals early on at the period corresponding to adolescence to 9-THC, you have an increased consumption of higher quantities of other drugs, like is shown here for nicotine, or actually also a higher risk for consumption of 9-THC, as shown to the right graph, indicating from the animal perspective that there is evidence that early exposure to cannabinoids, 9-THC marijuana, may prime your brain to make it more sensitive to the rewarding and addictive effects of other drugs. Very importantly, this study that was published in 2012 also addressed the other aspect, which has been one that has been questioned by those that believe that marijuana is a safe drug, and that ignore the prior finding that kids that smoke marijuana are not doing as well at school, that their cognitive performance is lower. That has been ignored because they say those kids, to start with, were already underperforming, and that's why they took marijuana, that's why they were more vulnerable. This study here actually challenged that, because in this study what they did was take into consideration the cognitive performance of those children before they got exposed to marijuana, and then follow them prospectively as they went into adulthood at age 32. And what this study showed was when you control for IQ, intelligence quotient, those kids that have continuously used marijuana during adolescence actually had a significant degradation of their cognitive abilities, as opposed to those children that did not consume marijuana. Independent, again, as I say, and I reiterate what their cognitive performance was before they got exposed to marijuana. I'm bringing forth evidence that the exposure to marijuana itself may be responsible for that deterioration on cognitive abilities. So what is it that we know neurobiologically about it? And this is one of the studies that I want to highlight, even though it's a study that was done retrospectively. And what do I mean by this? This is a study that evaluated the connections of the brain So as I say, the brain is a network, and it communicates at large distances and short distances. And with imaging technologies, we can actually measure the density of those connections. And this is what this study showed, where it compared the connections in individuals who had 59 individuals who had smoked extensively marijuana during adolescence versus those of controls that had not smoked marijuana. And in the colors there, you see the connections that differ significantly between these two groups and showed a marked decrease. And when I speak about a mark, I would say basically what is a mark? 80%, 80, fewer connections in those tracks that you observe there, which are fascinating because on the one hand, this is the track that connects with the hippocampus, which is the track that is involved with memory, and this could explain why there is a deterioration on the cognitive capacity of these children, because the connections that lead you to bring the information there are actually very much depleted. And the other area, which I found fascinating, is this one here, the precuneus. At one point in my life, I did not know what the precuneus was because it has not generated much attention like hippocampus or frontal cortex. The precuneus is the most connected area of the brain. It's an area that is fundamental for our capacity to create consciousness, for alertness, and that is profoundly disrupted by cannabinoids, as shown by this study. The younger you started to smoke marijuana, the less the connections of these two very important tracks in our brain, providing us evidence that the exposure to marijuana during adolescence may be impairing cognitive performance by interfering with the density of connections of key areas of the brain that are necessary for us to perform properly certain in cognitive tasks. But let's come to the adult brain, because as we are seeing, certainly in the United States, the greatest risks that we are observing are in those young adults that are 18 to 25. And I must say, we now know, that the human brain, in fact, does not fully form until we are in our mid-20s. So 18 to 25 years of age is a period in our life where our brain is still undergoing significant changes. But let's go beyond 25 years of age. Let's speak about the adult brain in general. Let's assume that it's already fully formed. What do we know? Can marijuana also affect it? Well, we know from studies in imaging that if you are smoking marijuana, your own endogenous cannabinoid system is going to undergo changes. And multiple animal experiments had already shown that exposure to marijuana in those animals leads to a reduction in the signaling through our endogenous cannabinoid system. And that's typical of biology. You stimulate one system beyond what is normally physiologically supposed to be stimulated, you downgrade it. That has been shown not just for the brain, but any other organ. And what you see here is the concentration of cannabinoid CB1 receptors, which are necessary for our endogenous cannabinoid system to function. And basically on almost every single area of the brain, when you compare controls versus marijuana abusers, you see a significant reduction in the levels of cannabinoid receptors. So what is the consequence? The consequence is that when these people are no longer going to be intoxicated, they are actually going to have reduced signaling through our endogenous cannabinoid system. This is a more recent study that does not look at cannabinoid receptors, but actually looks at one of the enzymes that regulates the intensity of signaling on the endogenous cannabinoid system. Again, this is very, very precisely regulated by synthesis and degradation. And in this case, you see one of the main enzymes involved with the degradation of the endogenous cannabinoid called anandamide. And you can see whether it's actually, this is the concentration at different times after administration of the radiotracer. This is, again, significantly downregulated in various areas of the brain of individuals that are marijuana abusers. So there is a downregulation of the cannabinoid receptors. There is a downregulation of the enzyme that degrades one of the main endocannabinoid systems. And these are the two ones that have been evaluated. One can predict that as we are able to evaluate other factors that mediate the function of this system, we will also replicate this finding. And this is a different type. This is not looking at the endogenous cannabinoid system, but looking at the dopaminergic system and different studies that have looked at it from different perspectives. And why is this important? It is important from two perspectives. One of them, the dopamine system, is what mediates the rewarding effects of drugs. So we want to know if marijuana influences the function of the dopaminergic system. But the dopaminergic system is not there for us to take drugs. The dopaminergic system is there for us to motivate our actions. So for example, right now, one of the most powerful behaviors, drivers for us as human creatures is social interactions. And yesterday we were speaking about tenderness and love. That's also part of the social infrastructure that is mediated by dopaminergic system that actually motivates our behaviors towards others and towards ourselves. The dopaminergic system is that one that is necessary for us when we wake up in the morning and look through excitement and drive and energy of what we want to do. If you degrade that dopaminergic system, which we can do in animals, you actually profoundly disrupt the motivation of those animals to do the most elemental behaviors to the extreme. The animals will not survive because they don't have the drive that motivates them to it. It's as basic and fundamental as that. And one of the issues that has been, certainly in my brain, of great concern is not just cognition. Cognition is very, very important. But in my brain, as important is motivation. And one of the things that people have recognized, and you are taught it in medical school, is one of the consequences of chronic use of marijuana is what we describe as amotivational syndrome. The lack of energy, the lack of drive, the disinterest on everyday activities that is likely to contribute to the dropping out of school. People say, I don't care to work, it doesn't really matter. Well, if we think about the consequences of reducing motivation in our young population, we can see why we need to pay attention to it. So these studies actually investigate whether marijuana is influencing that chemical system that is necessary for us to exert a drive. In this case, for example, the one to your right, you observe the synthesis of dopamine, the enzyme that synthesizes dopamine. And what this particular study done, which was published 2 years ago, was the lower the synthesis of, or the concentration of this enzyme that is necessary for produce dopamine, the greater the symptoms of a motivation. And there's two studies on the left, which are independent studies, one of them published in 2014 and the other one very recently in 2016. What they are doing is measuring the release of dopamine, how much dopamine is being released in these centers that are involved with motivation. And what the study in the upper part shows, which was a study that we did at Brookhaven, is that the lower the amount of dopamine, the more intense the negative symptoms. And what are negative symptoms? It's the negative aspect of our personality: anxiety, irritability, insomnia, distress, motivation, and anhedonia. And similar findings were reported recently by the group at Columbia that again showed that in marijuana users, the decreased release in dopamine is again associated with these traits that we've recognized with the repeated use of marijuana. So we now know that one of the consequences of marijuana in an adult brain, I'm not speaking of adolescence, is that decrease in motivation, which of course, when you are thinking about individuals professionally engaged, could explain why their productivity goes down. And I want to end up my presentation with marijuana and mental illness, because it's something that we need to also keep an eye about the potential negative effects of marijuana, and that has generated a lot of attention as it relates to, particularly I would say, two mental disorders. One of them is schizophrenia, and the other one is anxiety disorders. And I'm just going to be speaking about schizophrenia because, in my view, it's probably one of the most devastating mental illness. And if indeed the use of marijuana is associated with higher risk of schizophrenia, this is something that we have the obligation to communicate in an objective and way. When was this actually identified? This was identified, I would say, close to 30 years ago, and it was a study first published by Nancy Andreasen in a large cohort, 45,000 of Swedish conscripts. And what she found was that the increase of schizophrenia was significantly increased in those individuals that have smoked marijuana more than 50 times. Then that was then replicated by multiple studies, but I'm bringing this one, the Dunedin study in 1037, because this is a prospective study in which they show that indeed it increased the risk of psychosis, particularly if you use marijuana when you were less than 15 years of age. So here you show that the more frequent that you use it, the greater the risk. Here it shows that the younger you use it, the greater the risk. More recently, a group in England showed that the higher the risk, actually it's a very high risk, close to 6-fold higher risk of psychosis when you consume marijuana that has very, very high content of 9-THC. And as I say, that's one of the changes that we are observing. This is the active ingredient. And very importantly, I want to highlight the study to the right, the upper part of the quadrant, because I think this is where we're going to be uncovering a lot of the associations between drugs and disease, or between other environmental effects and disease. We know that genes are very important in schizophrenia. We know that genes are very important in addiction. But what is emerging as a result of the studies on marijuana and schizophrenia is that it can increase the likelihood of you developing schizophrenia if you have the genetic or the behavioral risks. What's a behavioral risk? If, as you are growing up, you are exposed to a very stressful environmental condition or a situation of deprivation. If you have the genes that make you more vulnerable and you get exposed to marijuana, then that may actually trigger the episodes of schizophrenia early on, or may trigger an episode that would have not happened, as shown by this study that I'm highlighting here. AKT1 is a gene associated with schizophrenia. And there is a risk allele that actually is described here as a CC allele. And you can see that definitively this individual has a much higher risk than those individuals that don't have the genetic risk. But what's interesting is if you do have the genetic risk, you will only increase the risk of schizophrenia if you smoke marijuana, but not even if you have the gene risk. If you do not smoke marijuana, that's not going to actually emerge as an increased risk for schizophrenia. Highlighting what I think is one of the most interesting components of our understanding of genetics. In most instances, having a gene that makes you vulnerable does not mean that you are predetermined to have that disease. What we know is that the risk that a gene imposes to you as an individual, in most instances, is by mediating the way that you respond to an environment. So if you have the risk in a gene and you're exposed to an adverse environment, then you develop the disease. But if you have the risk gene and you are in a protective environment, then you don't develop the disease. And as we think about drugs, this is something that we need to highlight. Because if you have the gene for a disease of a psychiatric illness, like is this situation like here, exposing yourself to marijuana may trigger a condition that otherwise would have not been possible. Why is that so? There's a lot of research trying to understand why the consumption of marijuana may make you vulnerable for schizophrenia. And I'm going to highlight one to my brain that is likely to be very, very contributory. A very consistent finding, study after study, has shown that the consumption of marijuana in adults is associated with volumetric morphological changes in two areas of the limbic brain that are very, very relevant, and it's one of the areas that's most sensitive to the effects of marijuana, the hippocampus and the amygdala. The hippocampus is important for memory, the amygdala for emotions. Study after study, you see that cannabis users have lower volumes. You see here in green. In this one, you see them here, smaller volumes. In schizophrenia, it also happens that this is a very consistent finding. The smaller the volume of your hippocampus and your amygdala, the greatest the severity of the symptoms of schizophrenia. Why is that so? Because the amygdala and the hippocampus regulate the way that the frontal cortex actually responds to stressors. So if you do not properly regulate that, you actually are not buffering the reactivity of your brain to those situations, which makes you vulnerable for psychosis. And this may be, in my brain, I'm hypothesizing because there are other findings that could mediate these effects, why smoking marijuana make you vulnerable. Is making the morphological, the functional changes of the brain that are making you vulnerable to stress and psychosis is worsening it, it's accelerating it. So how do we take this information in the current state of affairs of the world, and certainly in the United States, where policy is changing and making the belief that this drug it would be wonderful. If it was correct, it would be wonderful if we could sort of say, yes, marijuana is a safe drug. It would be fantastic. Unfortunately, that is not the case. And I like to show this one to test what are the consequences. I say marijuana among teenagers, and these are 17 to 18 years of age, has not gone up. But what you clearly see from 1994 to 2014, that it's basically stable. But it's stable at the time that the content of marijuana of 9-THC has increased threefold. So you have a much more potent drug that has nothing to do with the 1991. But we're seeing two parameters. Look at tobacco. So now kids in the United States, when you ask them, do you smoke? They think that you're speaking about marijuana, not tobacco. And for 4 years in a row, and we now have the data for 2015, 5 years in a row, marijuana, there's more frequent use of marijuana than tobacco. Now, that means something that we'll need to learn. We've been very, very effective at prevention of tobacco. And I brought this slide actually to sort of say, we've been able to bring down the consumption of tobacco. We've been very successful. Perfect? No, it's not perfect. We're still having a lot of kids smoking tobacco. But as we are moving forward into policies that are actually being driven by the interest to generate a marijuana industry, we should learn, not take 50 years like we did with tobacco, to prevent the adverse effects of consumption in marijuana, with a priority for teenagers, because they are the ones that are most vulnerable from many, many perspectives, but also, as I say, highlighting to potential negative consequences in the adult brain. And with that, I just want to actually advertise a large project that has become our main initiative, that right now, as an institute, the National Institute on Drug Abuse, in partnership with other institutes at the NIH, is actually launched a study last year in which we're going to be periodically measuring the brain of children as they transition into adulthood over a 10-year period. We're going to be evaluating 10,000 children in order to be able to unequivocally understand what is the normal variability of human brain development, number one, how does the environment influence it, and how do drugs affect it. And this information is, of course, very, very important, because if we can start to understand better how the drugs may interfere with the brain architecture, then, of course, we can do intervention to try to revert it. But very importantly, to use this information in a way that is unequivocal as it relates to our understanding about how drugs influence the human brain. Because teenagers, even though we say they are rebellious, they pay attention, just like we have seen that they pay attention to the adverse effects of smoking into our health. The challenge is how do we communicate this information in a way that is of value to them. And for that, we have to be clearly objective and scientific. Thanks very much.